Welcome, and thank you for joining our webinar, Accelerating AAV Vector Development with AI. My name is Angela Anderson. I'm the Senior Director of Strategic Growth at FormBio. And if you're not familiar, we provide computational and AI-focused solutions for faster and more predictable AAV development. I have my colleague, Dr. Anusha Sriraman, the lead for Life Sciences Insights at FormBio here today, and we'll be discussing how Resilience and FormBio teamed up to apply in silico techniques to accelerate vector design while also producing safer and more manufacturable vectors. We'll conduct a short Q&A at the end of the session, so if you have any questions during the webinar, please feel free to type them into the chat window at any time. The agenda today, we'll start out with a brief introduction, followed by an overview of the FormSight AI technology. We'll discuss the challenges in vector design, and then conclude with the Resilience case study. So first off, I'd like to briefly introduce FormBio. And just like you'd engage with a CRO or a CDMO, we think of ourselves as a contract computational organization that sits between the two. We serve as your dry lab partner for all your in silico needs, and our goal is to save your therapeutic programs years and millions on your way to IND. We're engaged with many leading companies in the CGT and AI space, including academic and commercial. We have a variety of strategic alliances, including PacBio as their hi-fi sequencing partner, and we're also honored to have a stellar scientific advisory board. So let's move on a little bit to the challenges. The promise of cell and gene therapies is undeniable, with their therapeutic reach expanding into earlier lines of treatment, across new indications, and to broader patient populations. However, we're seeing one of the biggest hurdles still is in the development process and ensuring that vectors are safe, effective, and manufacturable. So let's take a look at how traditional vector design works. We've come to realize that no two gene therapy developers operate the same. But what I'm sharing is a generalized example on our conversations and collaborations with hundreds of gene therapy professionals. Typically, just one vector design cycle can take 3 to 4 months, and you don't just do that once. You repeat the process several times. This iterative cycle of synthesis and in vitro testing isn't just painfully slow, but it's also incredibly expensive. And plus, you never know how many more design cycles you're going to need before you have a viable lead candidate. So that's why we teamed up with Resilience to investigate how AI technology could accelerate vector design and development. And spoiler alert, the answer is yes. In silico design can dramatically reduce time and costs by using AI and ML modeling to optimize and de-risk vector design. So ultimately, this enhances the safety, manufacturability, and probability of preclinical success for therapeutics. And now I'm going to turn it over to my colleague, Anusha. She's the one with the PhD who will break it down to you in much more detail. So take it away, Anusha.
Thank you, Angela. I'm excited to introduce FormSight AI, a cutting-edge AI/ML technology designed to solve key challenges in vector design, such as predicting truncated genomes and identifying problematic motifs. After outlining these challenges, we'll dive into the method and results from our collaboration with Resilience. Now that we're starting to explore FormSight AI, let's first focus on how this technology helps achieve specific design goals in vector design and optimization. These design goals that we have listed here are tailored to the therapeutic indication and gene of interest. So they're going to vary from one program to another. And let me walk you through how we approach these objectives. So the first design objectives is to reduce the truncation risk. By lowering the truncation risk, we minimize the number of capsids that lack the intended therapeutic payload. And we can achieve this by adjusting the minimized predicted truncations and the balanced GC content and CpG island knobs within our model. Some of the other design goals that we can explore include minimize immunotoxicity risk, wherein we can lower the CpG islands in the CDS to reduce the chance of triggering TLR9 immune recognition. Then we have enhanced protein expression, where we can improve the translation efficiency by optimizing the codon adaptation index. We can maintain wild-type gene expression by minimizing the codon substitutions within the CDS to preserve the native gene's characteristics. And lastly, we can also achieve tissue specificity by using a tissue-specific codon table to enhance protein expression in the target tissue. And so with these design objectives in mind, let's look at how FormBio's AI/ML engine optimizes the coding sequence to meet these goals. So FormBio's proprietary AI/ML model architecture enables the evaluation of these predefined set of objectives and constraints to achieve a specific biological objective. So once we have defined what these objectives are, the input CDS sequence is optimized based on these objectives and constraints. The optimization engine iterates through multiple combinations and produces an output that meets the set objectives that we've initially defined. And before we dive into how the model performs, it's essential to understand the kind of data it has been trained on. The quality and the diversity of the training corpus directly influences how well the model can predict and solve problems in real-world scenarios. So here is a representative training corpus, and this corpus contains sequencing data from multiple combinations of promoters, CDS, and poly-A-tail. So since FormSight AI has also been trained on an extensive list of extensive sequencing results from multiple gene therapy vectors, it can offer more accurate predictions, avoids overfitting to narrow cases, and also helps to identify patterns even in complex data sets. So this helps us to design vectors that aligns with your therapeutic goals. Now let's shift focus to how FormSight AI can specifically help with vector design. And let's start off by looking at some of the key challenges that arises during vector design and how FormSight AI can help address them. So some of the key challenges that we face during vector design include ensuring the vector design allows for the efficient packaging of the therapeutic gene into the viral capsid. This is very critical for a successful gene therapy delivery. Partially filled capsids can result in reduced transduction efficiency and also compromise therapeutic outcomes. Secondly, it is to identify any problematic structural motifs, because this can impact the vector's ability to be replicated and packaged into viral particles efficiently. Thirdly, we want to identify if the vector is prone to immunogenicity risk. So minimizing that risk is crucial so that we avoid any adverse events. Fourthly, we want to ensure that we can optimize the coding sequence so that we minimize the risk of premature truncation or termination, because that helps to maintain the functional integrity of the therapeutic gene and also enhances the therapeutic efficacy. And lastly, it is to accelerate the screening process so that we can identify the lead gene therapy vector. This is very crucial for expediting the development and translating the promising candidates into clinical applications. So in the following slides, we'll go into more details on how FormSight can mitigate these challenges using in silico predictions, and how we are able to de-risk the safety and manufacturability of gene therapy vectors. Okay, so now let's take a closer look on how FormSight AI predicts truncation risk, which is a key challenge in AAV vector design. So in this graph, on the x-axis, we have all the different vector elements listed. So in this case, we have a vector that contains the CAC promoter and the EGFP CDS. And on the y-axis, we have the read and counts. So this is actually indicating at which which region of the construct is where we can expect to see a lot of truncation read and counts. So in this case, our model is predicting that the promoter is a region where we expect to see a lot of read and counts, indicating that this is a very problematic region. And you can see that when you look at the forward replication sequence of this particular construct. Okay, let's move on to the next output that FormSight AI provides, which is to analyze the secondary and tertiary structural motifs. And these motifs are important because they could correlate with a replication fork stalling or other truncation events. So herein, we're actually examining the likelihood of a given set of nucleotides to fold into any complex structures like G quadruplexes or hairpins, because they can disrupt replication. Identifying these motifs allows us to understand if they are actually correlating with a truncation event. And so in this case, we see a lot of problematic structures in the promoter region. And given that they're shaded, it indicates that they're actually correlating with a predicted truncation event. In this slide, we're also showing you what are these motifs that are predicted within this particular vector. It's indicating, is it a G quadruplex? Is it inverted repeat? Is it an A-phase repeat? And how strongly correlated they are with the truncation event. So the lighter colors indicate that there's a very low correlation with the truncation event, and a stronger, darker color indicates that there's a very strong correlation. So this also helps you identify what is the exact motif that is predicted within that particular construct that you have. And now that we've covered the structural motifs, let's move on to another critical factor in vector design, which is the GC content and CPG islands, which are closely linked to immunotoxicity. So herein, we're also looking for what is the GC content across the construct, and do we identify any CPG islands? So in this case, we see that there are a couple of CPG islands indicated by the orange line throughout. And we see that there are these CPG islands present within the promoter and also downstream of the CDS. And so we are basically flagging them. Of course, in the promoter, these CPG islands are important for gene regulation. But when we see them within the CDS, we are flagging them for potential immune recognition by the TLR9 signaling pathway. So that's also the other information that we provide from FormSight AI. So now that we've discussed all these risk factors, let's look at how FormSight AI optimizes the coding sequence to reduce truncation risk. And so here is an example where we have the initial vector in purple dots and the FormSight optimized in pink dots. So we're specifically looking at the CDS here. In this case, it's mCherry, which is typically not known to have a lot of truncation. And so what you see here is that with FormSight optimization, that is indicated by the pink dotted lines, we are actually reducing the truncation. We're still able to reduce the truncation risk, and it's hovering at the noise floor, which is indicated by the black dashed line. So by reducing the likelihood of truncation in the coding sequence, we are not only enhancing the therapeutic efficacy of the gene, but also improving the manufacturability. And this helps to make the entire production process more efficient. And so this brings us to the broader impact of these optimizations, right? How FormSight AI reduces the number of trial and error cycles and helps to accelerate vector development. So by performing these in silico assessments, FormSight AI helps identify vector designs that are most likely to succeed. So with access to libraries of promoters and polyase sequences, we can test multiple combinations and narrow down the best candidates early in the process. And this significantly reduces the need for iterative trial and error cycles in the lab, helping to accelerate the development of your gene therapy drug substance. So in short, we are able to fast track the development timeline while maintaining a high degree of accuracy and predictability. Okay, so now that we've walked through how FormSight AI helps improve vector design, the next big question is, how does this work in the real world? So let's shift gears and take a look at the real world case study with resilience. So in this case, we investigated whether FormSight AI can be used to streamline the vector design and development process by predicting which genetic elements can perform better for manufacturing AAV vectors. The second thing was to optimize the coding sequence. In this case, the coding sequence was the human growth hormone to reduce the proportion of truncated genomes and test this optimized coding sequence, all of this, in Resilience's proprietary suspension AAV platform. And so the hypothesis that we had was that FormSite AI can accurately identify which vector elements would result in overall low truncation, so therefore improving the packaging and the manufacturability. And secondly, that when we use FormSite AI for optimizing the coding sequence, we would be reducing the overall truncation risk. So with that hypothesis in mind, this was our experimental design. So we evaluated two different promoters. In this case, it was CMV and JET. As I mentioned earlier, the CDS that we used here was the human growth hormone. So we used the initial unoptimized human growth hormone, as well as the FormSite optimized human growth hormone. So in total, we had 4 vectors that we evaluated for this particular experiment. Now, these vectors were packaged in a proprietary capsid and was manufactured in partnership with Resilience. We assayed for long read sequencing using PacBio, and we followed PacBio's best practices guide to ensure that the library prep, all of that was done according to their instructions. And finally, the results that we obtained for the long read sequencing data were analyzed using LAVA, which is the long read AAV analysis. It's an open source code that was developed by our AAV working group. So I've also linked, please check out the GitHub as well as the bioRxiv paper. I highly recommend that, which gives you details about the code as well as the process, the kind of outputs that it helps provide. So let's take a look at what are the key results from this study. So what we identified was that, yes, using FormSite AI, we can accurately identify the combination of vector elements that results in low truncations. And we are able to improve the manufacturability. And the second is that our FormSite optimization of the coding sequence is ameliorating truncation peaks. And this is actually able to improve the manufacturability of these constructs. And so with that in mind, let's move on to the detailed results. So firstly, of course, the objective was, can FormSite AI identify those vector elements, right? And so in order to test this, as I mentioned earlier, we have 2 different promoters, CMV and JET. And of course, the CDS and poly-A tail, all of that was kept constant. So let's look at the FormSite AI prediction. So in this case, on the x-axis, you see the 2 promoters, CMV and JET. And on the y-axis, we see the truncation propensity, which is how much truncation risk is contributed by each of these different vector elements on the y-axis. So in this case, our model predicts that the JET promoter is expected to have lower truncation propensity versus CMV, which indicated that JET is predicted to package better. It's expected to have lower truncations overall. And this is going to impact, the packaging efficiency of these vectors. So let's compare our predictions with the NGS results. So in this case, on the y-axis, we have the read end counts. And we have zoomed in on the enhancer promoter region. So in this case, we have the CMV enhancer on both constructs. On the top, we have the CMV promoter. And on the bottom, it's been replaced by the JET promoter. So what we see here is that in the CMV promoter, there are a lot of these peaks, right? Which is indicating that there are a lot of read end counts that are terminating at those locations. And so those are the truncation hotspots. And so these peaks are, we see a lot of these peaks within the CMV promoter, indicating that there's a lot of termination taking place at these locations. And when we look at the JET promoter in comparison, these peaks are absent, indicating that the promoter is not very problematic, right? We don't see those truncation hotspots. And so the JET promoter is definitely a better performing promoter in comparison to CMV for packaging. And so our model accurately predicted this, that the jet promoter is going to be the more superior promoter in terms of packaging. So yeah, FormSight AI was accurately able to identify this. Moving on to the second objective, it was to determine whether the optimization of the coding sequence using FormSight AI would actually reduce the number of truncated or partial genomes. And so in order to test this, let's first look at what the FormSight AI predictions were. So on the x-axis, we have two graphs here. So one is for the CMV promoter with the edge, and the other is for the jet promoter with edge. On the y-axis, we are showing you what the truncation propensity is, specifically at the CDS. And we have graphs for both the initial CDS as well as the optimized CDS. So the optimized CDS here is in pink and the initial CDS in purple. Now, regardless of the promoter, what we see is that when we've optimized the coding sequence, there is a reduction in truncation risk, right? So it's indicating that the optimization is expected to remove some truncation peaks, thereby helping in overall manufacturability. So we compared these predictions with actual NGS results, and here's what those look like. So in this case, we have the CMV edge and the jet edge, the initial CDS of these particular constructs laid on top. And then on the bottom, you see the CMV edge optimized and the jet edge optimized CDS. So when we compare the initial and the optimized coding sequences for both the CMV promoter and for the jet promoter, what we see is that there are a lot of these truncation peaks identified within the CDS of the initial coding sequence, regardless of the promoter. And these truncation peaks are ameliorated when we look at the optimized sequence. So this indicates that there's going to be, we would expect to see more full genomes, right, based on this particular finding. And that was actually true. We note that we see about 14 to 28% increase in full genomes when we optimize the coding sequence of CMV edge and jet edge respectively. And so with that, what I wanted to conclude is that FormBio's FormSight AI technology streamlined the AAV vector design process, and Resilience's innovative vector biomanufacturing platform produced and experimentally confirmed the FormSight AI predictions using long-read sequencing data. In addition to the increased genomes that we noted when we optimized the coding sequence, we also saw a twofold increase in genomic titer when we used the FormSight AI optimized human growth hormone coding sequence coupled with the better performing jet promoter. And so this genomic titer improvement was independent of other bioprocessing parameters. And in conclusion, what we can derive from this particular case study is that we can certainly use in silico tools, and these tools can help us accelerate the vector design and development process. And so as we near the conclusion of the webinar, I want to take this opportunity to extend my heartfelt thanks. Firstly, I'd like to express my deepest appreciation to everyone within FormBio. Your unwavering dedication, tireless efforts, and innovative thinking have been the cornerstone of our achievements. I also want to extend a special thank you to our esteemed collaborators at Resilience and UT Southwestern. Your partnership and commitment to excellence have been very pivotal in our journey. And the studies that we've undertaken together have not only enhanced our products, but also forged strong bonds and a sense of shared purpose. Finally, I also want to give a big shout out to our AAV working group. FormBio has worked with leading experts across academia and industry to develop an open source software solution called Lava, which is long-read AAV analysis. And this helps to simplify and standardize the process of analyzing and interpreting PacBio long-read sequencing data to get detailed characterization information on vector genome sizes and also residual DNA impurities. I'd like to thank each and every one of you for your valuable contributions, and a big thank you for everyone tuning in today for the webinar. We appreciate you taking the time. Before we jump into the Q&A, I also just wanted to point out to this QR code. If you scan this, it'll take you to a document where we've also done a couple of other experiments to test our FormSight AI model, and you can check out those results too. Yeah, and with that, we'd love to hear from you about your questions. Thank you.
All right. Yeah. Thank you, Anusha. We have a couple questions in the chat, and if you have any other questions, please feel free to type them in. So first off, how have FormSight AI optimized vectors expressed in vitro? Can you tell us a little bit about that?
Yeah, that's a very good question. So in another experiment where we had a readout for expression, which was basically using a protein activity assay, we found out that FormSight optimized vectors. We compared basically FormSight optimized vector versus open source codon optimized vector. And the readout was enzymatic activity assay. And what we saw was the FormSight AI optimized vectors did about 20%, like expressed 20% more than an open source codon optimized vector. So we did see that there was increased not only RNA, but also protein activity with our FormSight optimized vectors. And I also want to point out we had another study where we looked at GFP expression using FACS. And in this case, we found out that the expression, regardless of optimization, was very, very similar. So it was at par with the initial construct, but just improved packaging. So those are the two examples that come to mind when we talk about how FormSight AI vectors have expressed in vitro.
Okay, great. Thank you. Here's one other question. Can AI-based predictions be validated reproducibly? Like, for example, say you see a high correlation between predicted truncation and the NGS data.
Very good question again. And yes, we do see a high degree of correlation between our predicted truncation propensity as well as the NGS data. I encourage all of you to scan the QR code, because you'll see detailed results here about how this correlation, what is the correlation that we see across like 42 different vectors. And I also want to point out that this is, the case study that we presented today is also showing you a very good correlation, right? Whatever we see with our FormSight AI predictions is being validated by actual sequencing data. And so that gives us a lot of confidence that our model is able to identify, which regions are problematic and how we can mitigate those problems with our optimization.
Okay, great. Well, I think that wraps it up. Thank you everyone for joining and